All right, well, thanks for coming, everyone, today. So we have a visitor from University of Amsterdam. He's a PhD student there, and he works with an E.J. Wagenmacher's group, doing a lot of statistics for social sciences and psychology. We're really glad that he could come across the ocean to give us this primer on GASP, and he's also going to be working on incorporating some of our locally develop software into JASP this week, and there's a possibility we can maybe use this more in some of our instructional courses. So anyway, thanks for coming, and Frantisek, thanks for coming, and we look forward to your talk.
Yeah, thank you very much for having me here. And I need to just press on good it. It's a bit of a maze here because we have a very short HDMI cable, but we will manage. So welcome to the workshop on analyzing data with JASP, a free open source and user-friendly statistical software. And before I start, in case that you don't have a JASP installed in your computer, just go to the jaspstats.org, because part of this workshop will be practical. So if you want to follow and try yourself, it would make a lot of sense if you have JASP installed. Otherwise, I will also show how to do the analysis, but it would be nice if we could interact and you could also experience JASP firsthand. So just a very quick introduction about me. Who am I? I'm a PhD candidate of psychological methods at the University of Amsterdam. This is me on the picture. And I'm supervised by E.J. Wagenmakers and Dora Matske. And I mostly work on applications of Bayesian statistics, mostly in psychology, and I've done some work on meta-analysis, publication bias, and recently I also started doing some research on coin flips. Yes, there is yes, downloads, exactly. But I'm not going to be talking about any of these. I think I will have a talk about meta-analysis and publication bias later this week. So if you are interested, you can come and join. And I also contribute to JASP, and I developed mixed models, and I did a big part of the meta-analysis implementation in JASP and the learn-based module. You can contact me on Gmail if you have any questions that you forgot to ask during the workshop, and you can find me in 404D till Wednesday 15th this month. So just a very brief history of JASP, if you aren't familiar. JASP was started with my supervisor's grant called Base or Bust. And great, I have a first typo in the slides. And basically, the whole grant proposal was, all the idea was to create a way how to connect the theory of Bayesian statistics that was quite well developed to implementations in statistical software. Because in order to do Bayesian analyses, you would need to be, at that time in 2010, usually an R programmer or have some other very specialized software that's not very accessible and user-friendly. So the point of the goal of the grant and of the project was to bridge the gap between practice and theory of Bayesian statistics. And later on, we also added frequentist inference in a way to serve as a Trojan horse to the Bayesian statistics, because everybody always wants to do a t-test and ANOVA in frequentist settings, but nobody starts doing statistics and, oh, I want to do Bayesian ANOVA usually. And then adding those frequentist counterparts of the analysis to the JASP allows people to do both, compare the results, and slowly transition from frequentist to Bayesian statistics. And then also to work as a replacement for SPSS, Minitab, or other user-friendly software that's used in teaching statistics. So this is the plan, how it was supposed to work. So you have the JASP as the Trojan horse with Bayesian statistics inside, and that's how we plan to deliver the Bayesian goodness to the world. And since the project started in 2010, the team grew quite a lot. Now we have 3 core programmers that are employed at the University of Amsterdam. And then we have a lot of postdocs, PhDs, sometimes master students who do some projects in JASP as part of their master thesis or part of their PhD thesis. And all these people help the develop software. And recently, we also have people from Utrecht. So we have some programmers who help implementing software in JESP. And we also have a couple of programmers from eScience Center, which is an external software, science software support community in the Netherlands that help us develop the program. And not only we grew in numbers of the team that develops the software, but also in the universities that are actually using JESP for either teaching, analyzing data, or sometimes just hospitals that want to have a tool for to visualize data or analyze their data. Because you can imagine in many countries in the world, SPSS license is very costly and they just cannot afford to buy SPSS licenses for the whole department, hospital, etc. So it also helps to just make a more democratic access to education and statistical software. And we also have, that's not relevant for you, but for other people in the other parts of the world, we also have translation of JESP to Spanish, Chinese, and other languages. So you can also teach or use JESP in your native language. And since this transition, basically to be more global, we also want to we are slowly transitioning from just being supported by EJ's grants, because they will run out at some point, or most of them actually run out, to JESP and the development of JESP to be somewhat sponsored by the universities, because that allows us to keep the programmers running, keep the updates, etc. And we have a couple of universities in the Netherlands that already pledged to support JESP and that donate money or programmers to keep the project running. And we also had the A&M department here, University A&M in College Station, to kindly donate money and sponsor JESP. So in the end, basically JESP becomes more decentralized and more accessible software for all of us. So before we get to some practical illustrations how you can work with JESP, the core ideas of this JESP as of now as software is it's free and open source. It's user-friendly interface. So for example, in contrast to SPSS, etc., the user face is very interactive. It when you click something, it will change. You will see in a minute. And it has both frequentist and Bayesian analyses. And it creates visualization and reporting already output, which is also very nice. And it can be extended with additional analyses, modules, etc., that are being continuously developed. And it's available on both Mac, Windows, but also Linux. And if you start using JESP and you have some questions, then usually the best page where to start is at the JESP webpage that has all the links to all the relevant information. We also have GitHub where you can report issues, or you can ask for additional feature requests. And there is also a Cog Science forum where you can ask more conceptual questions. How should I interpret a base vector for interaction in ANOVA, etc. And it is also a backlog of the question that we answered. So sometimes you can find out the answers to your questions there. So now we can get to the practical part. So I will move to my computer because I will need to use the touchpad. And I have 3 examples prepared. So we will go through t-test and ANOVA. And then depending on the time, we can either look at beta analysis, or maybe better will be look at the learned stats and learned base module. That might be more relevant for people who are involved in teaching or demonstration of statistics. So for the t-test, I have an example data set called kitchen rolls, and it's based on the study by Topolinsky and Sparrenberg from 2012. Maybe just a disclosure, if you are from social sciences and you feel that I am being not fair to your field, I'm also from social sciences, so I'm also criticizing my field as well, so it's not personal. And Topolinsky and Sparrenberg in 2012 ran a study quite typical for social psychology, when they had a hypothesis that clockwise movement will introduce states of temporal progression. And orientation toward the future and novelty. So you can imagine the hypothesis is based that if you have a clock and you have the big hand, it's turning clockwise, and because it's turning to the future, so then if you look at the movement and you imagine the movement that's clockwise, then you are looking more forward to the future and you should be more open-minded to your experience. So it's part of the encoded cognition. Basically, like there's a hypothesis that the cognition influences your bodily states. And specifically in one of the experiments that they run, they had a study with 4 experiments, all supporting the hypothesis. And in one of them, they had 2 kitchen rolls. I will show you an image how it looked like. And they were turning either clockwise or counterclockwise, and they measured what's their openness to experience using a standardized psychological questionnaire. So this is the study that you can look up if you are interested. But this is basically the setup. As you see, so there are 2 handles, there are 2 kitchen rolls, they are wrapped in cellophane, so they don't unroll, and you are instructed to either turn them clockwise or counterclockwise. And what people in my lab did, they tried to replicate the study. So this is a picture from our settings when we have the 2 handles, the kitchen rolls with the cellophane, and we had 102 participants, which was 60% more than they had in the original study, and we tried to replicate the experiment and get the same results. And it will be the data set that we will be analyzing today. And in the original study, they found a t-value of around 2.3 or something with a p lower than 0.03, supporting their hypothesis. So, and we will reanalyze the data in JESP now. So I will just switch to JESP, and I will show you how you can load the data and how you can do frequentist analysis. And then I will leave it to you to try a Bayesian reanalysis of data set. And I will see if you have some questions, you can ask me for help, and we can see what's there possible to do in JESP. So I have a JESP open already. So this is what you see when you open JESP. It's the graphical user interface, and here is a top banner that shows all the analyses that are in JESP. And there is, I will just hide this. Right, yes, perfect. So these are the main analyses that are shipped with JESP. And also, if you click here on the plus button, you see additional modules that were developed in JESP. And we don't show all of them in the main ribbon because it would just make the interface much more messy. So there are different things like acceptance sampling, audit, meta-analysis, JAX, learn-based learn stats. We will look at some of them later if you have time. So to open dataset, we just click here on the burger menu, and we can go to open. I will show you how to open dataset from your local computer, but we also have this dataset in the data library. So just if you want to go to your computer, you'll browse, and then I need to find where is it in my presentations. I have the dataset. So I would just navigate, and then I would open it in the corresponding folder. So I would just open the kitchen rolls dataset. You don't have the dataset on your computers, but you can just easily open it by clicking on the menu. And if you click on open, there's also data library. In the data library, we have numerous datasets with examples and interpretation. So you can here, if you look at the t-test, there is a kitchen rolls dataset. And you should click on this upper part without the icon that just opens the CSV. If you click on the second on the left portion, it opens the annotated analysis already. So there are some saved analyses with explanation of the output and what we already shipped. So just click on here to open this specific data set. Then when the data set is open, you can see the data view. So there are different columns, and you can see the names of the columns. You can also change the type of the variable, whether it's scale, ordinal, or nominal, which influences how it's treated in analysis. There's also some textual variables that are treated as factors. And we can also do stuff like changing recoding of the factors, what the labels should be, how they should be ordered, or do filtering. So if we wanted to change whether we want to only participants with condition number lower than 50, we can select filters here, etc. We are not going to do that. We can also add additional columns by combining the columns we have. Or recently with the newer version, we also finally added data editing. So we could open the data set and we can edit the numbers directly in JESP, and we could change the values if we need to. Yes.
This app is the one you already downloaded to your laptop.
Yes.
This app works offline or online on your laptop.
This works offline.
Offline. Okay.
Yeah, and maybe one thing that's nice to know for teaching purposes, especially if you don't have admin rights to the computer you are teaching, we also have, for at least for Windows, we have a pre-installed version. So if we go to JESP and you click on download JESP, then you can either download here Windows to download the pre-installed version, or there is a pre-installed version that you don't need to install and you don't need administrative rights. It just has a zip archive that needs to be unzipped, and you can run just without installation and without the need for permission.
Is this similar to RStudio?
Not really, because this has I will show you now. If we go back to the analysis and we open the t-test, then let's analyze data with the independent samples t-test. It opens the graphical user interface for doing the analysis. So you don't really need to use any commands or any programming language to run the analysis, which is the main point of JESP, to make it very accessible and very interactive. But maybe before we start with the analysis itself, we should look at some descriptives, how the data look like. So we can just click on descriptives here, and we'll use the mean neo, which is the dependent variable, which is the mean of the openness to experience questionnaire based on the neo. It's the 5-factor model. It's from Big 5. And if you just plug it in, you see some basic description. What's the mean, how many values we have for standard deviation, and we can also create some basic figures. For example, you can look how the distribution of the data look like. So this should create a histogram, and then we can change what's the type of the bins, etc. Display density, display rug marks, etc. to just see how the data look like. This is not particularly nice or interesting plot because the data are just this oddly shaped pile, but we can analyze the data. So we can look, we can actually, we have the condition. So we can split data by condition, and we can see how does the distribution differ between the conditions. Did I split it by condition? No, so sorry, it should be with rotation. It's either clockwise, counterclockwise, yes.
Not any coding needed.
No, there's absolutely no coding needed. So basically the point is to replace SPSS, if we are familiar, that's usually used in social sciences for teaching. But the whole process is much more interactive, because in SPSS, you always have the output that's being generated, and you have to go back and forth. And this has the left menu where you have the whole panel with the controls, and the right menu that generates the output. And we can also copy the output directly. For example, let's say that we want to copy this image, and we can copy it to Word. This. And we can easily copy the images, or we can save them as PDFs if we want to use them in Overleaf or LaTeX software. And similarly, this works with tables. So yeah, just finish.
So far, you are using the data set already already in the software.
Yes.
So what if you want to use your own data set?
You can just load it from a file. So if you click on the open, and you can just search a file that you want to open, and you can open any CSV file here. And yeah, so we can also copy the tables out. So it's very easy to create already formatted output for all this. So if we just move to the t-test, then here we want to use the rotation, which shows whether the participants are in the clockwise or counterclockwise in the data set. And we can use the mean score as a dependent variable here. And immediately you will see, I will hide this. Okay, I will just close the descriptive so they are not interfering with our output. And immediately you will interactively get the result of the t-test, which shows the negative t-value, degrees of freedom, the p-value, and there is plenty of other options that you might want to explore. For example, whether you want to use a Welch t-test, you can just click on the button and it automatically adds the outputs for the Welch t-test, similar for the Manvit NAU test. And you can also specify whether you want to test directional hypothesis or not. I now don't remember which group was which, so maybe I will just look quickly at the visualization of the data. So we have the two conditions with the clockwise and the counterclockwise. And you see, if you just look at distribution, that the green one is slightly lower than the orange one. So the clockwise is turning this way, so they should be supposed to be more forward-looking. But yeah, this actually here, that's going to be more. So if you look at the descriptives, then we see with 48 participants in the clockwise, 54 in the counterclockwise, and the people in the clockwise have lower mean actually than the people in the counterclockwise. This difference is not statistically significant, but it also seems to be kind of in opposite direction than the original hypothesis was predicting. So the replication probably wouldn't support the hypothesis. The question is whether the data support the evidence of absence or evidence for presence, because we just have non-significant result if we use the frequentist analysis. And for that, we can continue with the Bayesian reanalysis of the data, and we can quantify whether there is evidence for the null hypothesis of no difference, or whether we don't know and we need to collect more data to actually collect evidence for there being a difference between the two conditions. So if you have your laptops open and you have the data set, you can now open the Bayesian version of the independent samples t-test. And you can try repeating the analysis and specifying in a Bayesian way, like evaluating the evidence. And I will give you, I don't know, 3 to 5 minutes. So you can just play around, see what the different options allow you to do. And then we can communicate the results and see whether what you found and what would be your conclusions based on the analysis. And I can also answer some questions in the meantime regarding the output that I presented so far. Yes.
While that's happening, what is the software that you're using to like generate the plots that we saw here?
Yeah. So I maybe should have mentioned it before. The Jess, all the background is running on R.
Okay.
So
Like it looks
yeah. No. Okay, so well, we have the 5 minutes to figure out. Hopefully it will just replug and it will work again. And oh no. So you have the front end, which is running QML, which is similar to JavaScript. and it's running R on the background. And all of the analyses are implemented in R. The code is very similar to base R, but we have some additional functions that allow us to forward output from JESP from R to the QML interface where it's rendered. So we have some formatting for tables, for figures. All the figures need to be in ggplot, so we can display them and we can modify them on the go. And we are relying on many JESP package on many R packages in JESP. So I think the t-test implementation is actually all custom written, so that's not part of a package. But for example, for meta-analysis, we have to rely on Metaphor for all the frequency stuff. If we use for the mixed models, they are running on LME4 and on Apex, which is nice for visualizing experimental data. Yes.
One related question, for this study, did they record handedness?
Yes, so they recorded handiness, and in the original study they found it did not matter. I don't remember in the replication whether it mattered. We can actually test that. So let's see when the people are finished with this question, we can quickly run a linear regression, and we can check whether handedness modified the result. But I remember that the original study they had to also adjust for two more variables to get the significant t-score, which is a bit also indicative of data dredging. Yeah.
I missed, what is the response that you are comparing?
Oh yeah, the response. Are you familiar with Big Five personality questionnaire? Okay, so it's a score on openness to experience from Big Five. So from 10 items, it's the mean score from that. Is there anyone still playing around with the Bayesian version of the analysis? Or should I just go on?
About
Yeah.
the code, like the programming behind this app, is this by R or by C++ or whatever?
I'm not sure whether I understand correctly, but yes.
It's like the backstage is C++ supporting.
Some packages that are also running, like some R packages also use C++ on the background. So for those, it's also running the C++ routines behind, but it's completely contained in the whole JESP. So if you install JESP, you don't need to install R tools or RCCP, etc. Everything is in the package.
The open developing app stage, or it's like a clone. For example, if other developers they want to contribute to this app, can they just freely participate or not?
Yeah, the whole code is open source and it's on GitHub, and we have many people who contribute to the project. And also, if you want to develop and add your analysis, you can create your own module. It's usually good to contact the team to include in the official distribution of JESP. And we also have a guideline how to start, what are the programming rules, yeah, like how should the code be formatted, etc. When you want to contribute to the if you want your code to be part of the distribution. But you can also make your package on the site, and you can sideload your modules into the JESP through this menu where if you have your own module that you don't want to be shared, theoretically you can also have module that's not available to anyone else. If you work for a private company and you want to have some specific analyses to just run for the people there, but you don't want the code to be publicly available, you can have your own tarball that you can just install here from developer module. So you can also install additional modules and expansions to JESP locally that will run in the R and in the background.
But there there's many algorithms here already.
Uh-huh.
Let's see the BSTS. Is this the Bayesian structure time series model or what this stands for?
I think it is. I don't know. Honestly, I'm not involved with all of the modules, and it's sometimes hard for me to keep track with everything that we have. But if you open it, no, it's state space. Yes. But yeah, there's a lot of modules that are being developed, and we do many of them in-house, but also we have people that wanna, for example, we had some exploratory modeling that people wanted to include into JESP, contacted us. We provided them with all the tools and resources, how to program in JESP and how to develop those tools. So if we just look back to the example in the Bayesian analysis. So the nice thing is that the Bayesian analysis works very similar to the frequentist one. So you just, again, find the variable, which is the mean score for the NEO, and put it into dependent variable, and you can select the rotation as the grouping variable. And then you automatically get, this is usually working immediately. The connection is probably not the best, and my computer for some reason doesn't like demonstrations. You immediately see the results with the Bayes factor against the null hypothesis. And maybe it's good to also look at the prior and posterior distribution to see how our prior distribution for the t-test looks. Here we use the default Cauchy distribution that was advocated in psychology, which is the local prior distribution with a scale of 707, which we can also verify here when we click at the prior tab, and we see the selection of the prior distributions. So, and you can then modify the scale of distribution, or if you want a different prior distribution, you can specify an informed prior distribution, and you can modify the location, the scale, or you can specify different prior distribution, and you immediately get a different result. The thing is also that the hypothesis wasn't whether there's a difference between the rotation, but whether the people who move clockwise have show more openness than the counterclockwise. So we should specify we should specify ordered. The test is not about equality, it's not about inequality, but it's about whether one group is larger than the second one. So if I remember correctly, the group 1, the test is whether the group 1 is larger than the group 2, which is by the rotation. And yeah, luckily we have a description under the table that says the location of group clock is greater than the location of group counter. So this is in the direction that we wanted. And we can see that if we also specify the direction of the hypothesis, then we get a Bayes factor about 0.129 in favor of the alternative hypothesis. There is the positive effect. That's not so super easy to interpret, so we can just quickly switch it to Bayes factor 1.0 to see what's the support for the null hypothesis of no difference versus the positive difference, and we find a Bayes factor of 7.75, which is medium evidence, let's say, in support of the null hypothesis of no difference under this prior distribution that we selected. And when we get back the video connection, we can also look, for example, on robustness check that shows how the Bayes factor changes based on the scale of the prior distribution. So we can see that when the figure loads, then we will see changing scale of the Cauchy distribution and how the Bayes factor changes as a function of Cauchy distribution. Yes, okay. So you can see that if you are increasing the width of the Cauchy prior distribution, you will get less and less evidence for, well, you get more and more evidence for the null hypothesis. And if you specify the width to be exactly null, 0, then there is no difference between the null and alternative hypothesis, and you will get the same evidence for no alternative. And whether you choose the default prior or wider prior, then the evidence is pretty much the same and doesn't really change. So you can also verify how robust are your results to the specification of prior distribution. So this would be the independent samples t-test. And then the question was whether the handedness plays a role. So we can just quickly check whether we have the variable here. I'm not sure. I know that the original study had it, but we don't seem to have it, so we cannot look into that. Okay, well, I'm not able to answer your question, unfortunately. But we can look in a second example of analysis that's very often used, and that's ANOVA. And in this data set, or in this example, we have data from University of Melbourne that was testing whether there is a association between a hair color and a pain. And there's a hypothesis there is some gene. I'm not a biologist, so if I mess this up, but there is some gene expression that modulates your ability to tolerate pain. And people who have lighter hair color has the same expression. The hair color is somewhat affected by the same gene that modulates your pain response. So you can use your hair color as an indicator for your pain tolerance. So what they did, they got a lot of people, well, not that much, couple of people with different hair color, and they caused them a lot of pain. And they're measuring how much pain did they tolerate it on different standardized pain stimuli. And the data are available at the department or on the department website. And yeah, so we can load the data in JESP. And if I just go back, is there something else I should say? Oh yeah, that's already said. So they had different stimuli that's used for pain research. People indicated whether they are painful or not, and they got some mean pain threshold score that we can use as dependent variable. And we can compare now whether there is a difference between different conditions according to the hair color. Oh, yeah, I see. Now it's okay? Yeah, sorry. Well, we will switch to JESP again, and we can just open the second data set that's also in the data library. So we go to open, data library, ANOVA, and then we find which was pain thresholds data set. So we just open it like this. It should start a new JESP window with the data, and we have the hair color variable. We apparently have 19 participants and different pain tolerance scores. So here we can open ANOVA, and in the classical statistics, we would just select the pain tolerance as dependent variable and the hair color as the fixed factor. If you are running the analysis on your computer, then hopefully you see that this doesn't take ages as here. And you get the default ANOVA output that you would get for the analysis again, which actually shows there is significant difference amongst the hair color levels. And we should probably display some descriptive statistics again to see the means and the different categories. And there should be some visualization again of different levels that will help us to understand the data. So we can look at the different grouping of the response variable by the hair color. And we just stretch this a bit so we can fit the different levels. And it seems like that the data are in accordance also with the hypothesis. When you have the light blonde people with the light blonde, which has the lightest color, have the highest pain tolerance. And we have people with dark blonde, which is the second level, like the second lightest hair color, then light brunette and the dark brunette. And these levels are somewhat ordered according to descriptive statistics. And then we can do all the different ANOVA tests and analyses that you might be interested in doing. So for example, you can specify different contrasts. If we had more factors, we could test the models hierarchically. You can look at the marginal mean, simple effects, etc. I think here we can just quickly run the Bayesian analysis to just verify the result again and see that trying the Bayesian version of the analysis as seamless as in the previous way. So again, we just need to specify the pain tolerance and the hair color. And we get immediately the same result when we can test for the presence of the effect by hair color. And here we get the base factor of 11.9 for the presence for the difference by hair color. The thing is, maybe one interesting thing that we can do with one of the added modules here is that the hypothesis originally was not that there's a difference, but that the lighter hair have more pain tolerance. And for this, we can use the Bayes module that I will just show you how to load in a second. And that allows you to also specify ordered constraint. So we can test the Bayesian hypothesis, whether, and we can figure out how much evidence there is for the hypothesis that the lighter the hair color, the more pain tolerance. So,
So what was the default alternative?
The default is that there is no difference between the levels.
So you have the same prior distribution on the level of each, or how is it?
Yeah, in ANOVA, there is we use the default JSZ prior, which has orthonormal contrasts. So basically a difference from the grand mean for each level. And that's also a good question that leads to how the analyses are specified, because it's sometimes not straightforward to just look at it from the options. Although I think here it's in additional options. We can specify the prior distributions for the priors, which is the R scale for the fixed effect and random effects, but also if you have multiple models, multiple factors, the analysis automatically model averages across the model space, and we can specify what are the prior model probabilities for different model complexity. But for this and for the implementation information, there's the I button, which shows information for the analysis that also has description, what are the options and how they work, which is actually quite important knowledge that I should have taught you before. So here you can see what are in majority of the analysis, there's description, what are the assumption of the analysis, and then how the different menus work. So what are the assignment boxes, how you can change, what are the options in the base factors, what they do, and also the output. Apparently there is also, we now have GIFs. I have never seen this before. Apparently I missed some updates. Oh, okay. So apparently you can also have live illustrations of the output. And at the bottom is resources to the articles that link to the implementation and more background knowledge about the implemented analyses. This also, maybe one thing that I should have told here is that if you have the analysis, you can easily annotate the analysis. So if you want to use them for your students to show them how the results should be done, etc., you can add a note here, and you can write, for example, that this is default output, and you can write some description of the different tables. You can also insert images. You can insert links to additional resources, and you can add the notes for each of the outputs. So this allows you to customize the output, and then you can save it and save the whole JS file with the notes, and you can send it forward. If you are familiar with OSF, which is a platform for sharing code and data, it can also display the JESP file is there. So if you do your analysis and save it and upload it to OSF, it's automatically rendered on the OSF page. So you don't need to download JESP and look at the results. You immediately see what has been done, including the comments. Yeah, but maybe the one need the demonstration of the different levels here is from the Bane package that's implemented in JESP that allows to test ordinal constraints on different variables. So for ANOVA, there's a Bane implementation of ANOVA, which is slightly different because it uses interesting base factors. So it uses the data to define the prior distribution, which allows then very easy specification of the different constraints. So for example, if we specify dependent variable as the pain tolerance and hair color, then we can, is it pain or? Oh, and we get an error message that tells us that doesn't factorials cannot contain spaces. And that will be because of the specification. So there is a way how to change it. I might have done it before, and now I forgot. Okay. We have still 20 minutes. Maybe I'm just thinking whether I should go through this now. Oh yeah, here it is. Yeah, we need to change the label. So we can just quickly edit the labels, which you need to double click on the variables of the column. And on the page, you will also see on the JESP page, there's information how to do all of these changes, how to modify dataset with tutorials and help. Okay, so now this should be updated, and we should be able to go back to the analysis. Okay, and now analysis runs without an error because we have the different factorial specified. And we can specify the constraint on different models. So we can specify a model that actually specifies the light blonde should have higher mean than the dark blonde, etc. So if we just write the names, which is hair color and light blonde is larger than hair color dark blonde, and we press it. I will just try. Okay, so I have the specification correct. You just need to write the factor name, which is the hair color, and then the level of the different factor, and then we can specify the whole list of ordinal constraints for different means, and we can test this order hypothesis directly. So the next one would be hair color light brunette. I have no idea how it's spelled. Probably, and hair color dark brunette. And I have apparently made an error in spelling. Oh, hair color. Okay, I should have written hair color here. And that's also part of the software is we are trying to make the error messages as informative so the users, if they mess up something, can fix the output themselves. As you saw, most of the menus so far were usually drop downs and checkboxes, which basically prevent user error. In those bit more advanced analysis, when you specify order constraints, etc., then sometimes the menu needs to be done in a way that users write down what is the specification, and then these errors can easily occur. So now when I update the menu, did I fix the error? Or is there still spelling? Our name, dark blonde, brunette. Okay. I don't now see what's my spelling error anymore. Light. Names in there. Oh, thank you. It also helps to be a native speaker to check your spelling. As you can see, my English spelling is absolutely horrible. That's why I do stats and not literature. And we can compare the base factor here in the matrix, and you see the comparison of this specified hypothesis against an unrestricted hypothesis, which is the U, which is also described in the note below the table. And the unrestricted hypothesis is that there is no ordering between the factors. So not only we find we find evidence in favor of the hypothesis that there is a difference between the factors, but we also find evidence for the ordering that's proposed by the hypothesis. And in fact, the data are around 18 times more likely under the model that assumes there is ordering corresponding to the theory that lighter hair color have higher pain tolerance. And yeah, so this would be like the two basic examples of analysis that you can do easily with JESP. And I can still quickly demo you some of the teaching tools that we have in JESP, because it might be quite relevant for the people here. So we can look in into the learn stats on learn base module, and we can just quickly check one analysis from each and see what's there. So I will close the analysis that we did so far. And yeah, this data view, I should just close this, and we can open the learn stats module. I will also prepare the learn base module that we will look into later. And these two modules don't require any data, so we don't need this data loaded, and they will be still available even without data set. And this module is still under development, so there's still some features missing, but we can, for example, look at p-values. And so when we are teaching p-values, there are some different default outputs that we want to display and visualize, and have some interactive features for students. And as you see, that we have some modules on different distributions, central limit theorem, standard errors, effect sizes, decision trees, but we are still adding mostly some explanatory text for those modules. So then you can just click and you will get default descriptions, what's happening where and what. But if you just go to the p-values, and we can look on some theoretical distribution of the test statistics. For example, we just use the normal distribution, so we would have z statistics. And when we get our visuals back, I think then we can display the main different things. For example, the critical region. So directly, if we specify two-sided type 1 error rate at alpha 5%, we can see the visualization, which test statistics would lead to the rejection of the null hypothesis, and how we account the 5% that's split in 2.5% into each tail. If we want to do one-sided test, then we can specify, for example, greater, and we can see the one-sided region for rejection. And we can specify, for example, some test statistics that we observed, and was the p-value of the corresponding test statistics, and was the data that would be such and more extreme under our test, given the test statistic. These are the very basic visualizations that you would have, but we can also simulate to show how the sampling distributions of the statistic work and look like. So, for example, under the null hypothesis, we can simulate one data point, and we can plot the test statistics, how they look like. And if we just simulate randomly one test statistic, it seems to be around 0.03 or something. And this is not retelling, so we can see how the results change if we add more test statistics. So this would add additional one test statistic, and slowly we would see the sampling variability of the test statistics under null hypothesis. And we can also plot them as p-values. And So we'll have the distribution of test statistics, of the p-values, and these are the corresponding p-values that we observe. And we can, for example, add additional 98 test statistics. So we have 100 of them. And now it will add test. Did I click it? Yes. And then you see what's the distribution if you perform the same test on a null hypothesis. And you will also see that you will observe some false positives of the test, which you can, and that the distribution still overall is somewhat uniform. And we can look at the frequency of p-values, how many test statistics you see that are significant and non-significant. And in this case, we have 4 significant and 94 non-significant. And I pressed it twice and I refreshed the whole analysis. So we have 98 instead of 100. And then we do the comparison. For example, if you look on the alternative hypothesis. So here we can specify the mean of the sampling distribution, for example, 1. It should give us an around 40% power to reject the null hypothesis. And we can simulate, let's say, 500 test statistics directly. And we could simulate. And we should also upload them. Like I'm using power. No, I'm running on battery. Okay. And then we get a simulation from the alternative hypothesis when we can again see distribution of the test statistics, of the p-values, and we could also look at the corresponding. Oh, thank you. Yeah. So we have the distribution here, and we can also plot the p-values. And see the distribution is right skewed with most statistics being significant. And we can look at the frequency that will tell us that I hopefully remember correctly that the test will have around 30% power to reject the null hypothesis with the distance and neutrality parameter. And then you can use those to in class do demonstrations and quickly get the output and play around with students or let them do some of the stuff. That's the teaching module for the standard for the classical statistics. And then one that I also worked on was the learned base module that we use when we are teaching Bayesian statistics at our department. So EJ is too lazy to prepare his slides and to always draw stuff for the students. So I had to make all the visualizations that he uses in JESP, so he can just come and play around and don't have to do anything. Don't tell your supervisors because they might want you to do that, do the same. And we can, for example, look at Bayesian estimation. So when we teach students, we try to do it interactively and get some data from them. So let's say that we can talk about our preference for cats and dogs, and we want to estimate what's the proportion of people that like dogs or cats. So let's first specify some hypothesis or like some prior distribution on the proportion. Let's look at some proportion. Let's try to estimate the parameter first and then test which one of those prior distribution is better. So is there anyone who has a prior distribution on the affinity towards cats versus dogs? Okay.
40% of people like cats better.
20%. Okay, so that's a spike at 0.20 would be the valence prior distribution. So I will just call it val. And I will just use the boring uniform prior distribution, which is the beta 1,1. And we can just immediately visualize our expectations, how the different prior distributions look like. So this is the spike at 0.2, which is the 20% people prefer cats, and I would just have this very wide prior distribution. And then we could just collect data in the classroom, and we can use C for cats, D for dogs, because we don't want to write all the names. So what's your favorite animal?
I'll go cat.
Okay, so we have one cat. What's yours? We have one dog person. We have another dog person. That's not an option. Either cats or dogs.
Dog.
Yes.
Dog.
Okay, we have one dog. We have a cat. We have another cat. We have a dog. We have a cat. We have a cat. I think Val will lose. We have a cat, and your data don't count because they are motivated. So we have the two levels, which is the cats and dogs. I will select cats as successes for no reason whatsoever. And we can look when we just estimate, when we use the data to estimate the models, how the posteriors look like after we collect the data. And as you hear in the model summary, the Wall's prior distribution was spiked at 0.2, so he couldn't learn anything. So even after observing the data that consisted of 6 cats and 4 dogs, he still thinks 20% of people like cats and 80% of people like dogs. But my prior distribution updated to a beta 7.5 posterior distribution, and we can then visualize the posterior distribution after observing the data. So how we learned and how the data updated our priors. And we can do different things. For example, we like to show the stack distribution plot that shows how the prior distribution changes with observations. And here you see that the spike is still at the same place, and the additional observations didn't do. Oh, just thinking whether it's. And while the beta prior distribution is updating according to the beta updating rule, and then we can, for the students, we can also make this obvious when we show the posterior updating table that shows how the beta changes with observation of the first cat, then a dog, etc. And how our posterior distribution after one cat is our prior distribution for our second observation, etc. And we can do many summaries. We can also look at what are the different credible intervals at the different levels. So how the posterior probability that, for example, that the value is somewhere between 0.2, 0.5 changes with the more data, etc. And we can predict what would be our expectation after observing those data for another 10 observations if we had a different classroom. So here we can look for 10 future trials, for example, and we look at the prediction for both hypotheses. And here we have the prediction for another 10 observations after observing the data. So I would predict that I would see mostly around between 2 and 9 successes. And that Val would expect mostly around the proportion of 0.2. Yeah, you see it now, the posterior prediction distribution. And if you look at the individual ones, we could look, was the point estimate and was the prediction interval for different distributions. So this will just quickly update. And we have the predicted number of successes under the Valls model, which predicts that we'll see mostly 0 to 5 cats, people in the next 10 observation, and under my model, where the prediction is 2 to 9. So I think And similarly, we can do similar analysis for Bayesian testing when we can specify two different prior distributions. Okay. Yeah, I might have played with the table a bit too much. So if you just copy the data quickly to the binomial testing, the base binomial testing, and we can enter the sequence again here with the cats and dogs, and we specify the same models. So just what we saw before, which will hopefully show when it's done. Then we can do, and for example, look how our evidence accumulates with the data. So we can look at the sequential analysis, and we can, for example, look how the base vector evolves with the observations that we see. And because we have only two hypotheses, that's that we can have either base vector in favor of my model or in favor of Val's model. And we could see on the figure that the evidence flows slightly more towards my model with the more data that we have. Okay, maybe we can end here. Yeah. Okay. Yes. It takes CSV files. We don't support Excels because to load Excels, I think there's some proprietary thing that you need to have. Maybe I'm not sure, but you just can save it as a CSV file, and the CSV files can be loaded into just directly. Yes.
And once I have data in JAS, how flexible is it? Like, can I make new columns out of previous ones?
Yeah.
Delete rows, add rows, whatever.
Yeah. So you can create new columns from the previous ones. You can also just create a new column as you would in a spreadsheet and enter the values. And when you create new columns, there are different ways how you can do it. There's some very simple drag and drop interface where you can sum different columns, take a ratio, take a sinus of one column, or you can also use R commands to specify more complex transformations of different columns. So if you know the column names, you can specify whatever you want. You can compute some complex effect sizes, etc., from the data that you have and the different columns.
So far, this app is free.
Yes.
Do you think in the future it can be like a charge some fee, or how your team to make a profit?
It's non-profit enterprise, and the JESP will be forever free. We have some companies that want us to develop some additional features for them, and I'm not completely sure because this is I think so far we were most developing programs for them if they are paying for the programmers to do it, and they allow us to make it available for other people as well. So basically, we sometimes have external companies wanting to do state space models because they control, for example, it's a glass making company, and they are measuring whether the glass is still in spec. So they wanted some specific models for that. So they paid for programmers to implement that into JESP, and that's also sometimes how we manage to raise more money to support the program. But the JESP will and is free, so that's not going to change. It's good to have some additional funding from different agencies or different universities, and as you saw, we had 5 universities that signed up in the Netherlands directly to support JESP in some long-term future. And even in the Netherlands, now the plan is to completely switch the education from SPSS, etc., in the social sciences. And all those departments are also changing what software they use for teaching. So instead of paying hundreds of thousands of dollars for SPSS licenses, they contribute a bit to the JESP Foundation which is then used to pay different programmers and the software development. Yes.
Question closer to physics.
Yes.
So, say you have a function,
Mhm.
one very,
Yes.
and that function has a certain number of parameters.
Okay.
Then you take the data.
Mhm.
You measure this function as this, function of this variable.
Yes.
Are there fitting tools that allow you to estimate
Mhm.
the parameters of this function, right? Say,
Yeah.
five parameters, and estimate the variance and uncertainties for those parameters
Yeah.
based on your measure.
Yeah, so this is a bit advanced topic that usually JESP would be used for, but actually we also have a JEX implementation in JESP. So if you can specify the model, your function in JEX, then I don't want to I will just check whether we have some example. In the data library, open. Is there an example with JEX? I don't see any. Well, you can, if you are familiar with statistical modeling language S10 or JEX, there is we have a JEX interface here that allows you to load in the data and specify. You can write a bit of and maybe it will load in a second. So I don't have any data now, but you can write that A is distributed as D norm. I think it's 0, 1, and so this is not really your specific model, but you can imagine you had some data that you would put into JESP. Then you can connect them to the model, and you can also write down stochastic models that are estimated with JEX, and you can get the summaries for the posterior distributions. You can get some visualizations, trace plots, etc. But I think it's possible, and it's definitely doable. To be fair, if I want to write or research paper on some complex model with has different measurements, etc., I would probably still use R because it just gives you much more flexibility. But if you want to use it for teaching demonstration and you have the models pre-written and you want to add annotation and have few variables that people can change and get the results immediately, for that it might be it would work well. But also there are different tools for different tasks, right? This is not like JESP is not supposed to eliminate R or be to be as allow you as much customization, because that's the strength and also the weakness of R. You can do basically whatever you want with it because it's a full-fledged programming language, but it also means that there is some cost of entering the ecosystem. And sometimes when you want to teach statistics, you don't want to teach programming side by side.
Let's take a speaker.
Yeah, and if you have more questions, I had my email there, or you can just stop by and I will try to answer or ask people who know more about to answer your questions. Thank you.